Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and salutations to all. I'm Muhammad Hakim bin Abdul Razak, and on this wonderful day, I'm going to present my review on the antimicrobial activity of Psidium guajava leaf, or commonly known as guava. Firstly, I will enlighten you with the purpose and background behind this research. As you all may or may not know, antibiotic resistance among pathogenic bacteria has been an issue and has been recognized as a challenge to public health for several decades now. This is the result of our own negligence of abusing and irrational use of antibiotics. This led to the huge interest in plant-derived pharmaceutical as an alternative to antibiotics. One such example would be guava. Guava is a fruit that is commonly utilized as food as well as traditional medicine. Various parts of the plant have medicinal values. Referring back to the main topic, in the crude extract of the leaf, we could find a huge variety of secondary metabolites that can be used as resistant modifying agents. In general, the secondary metabolite could be used as antibiotics, antimicrobial, as well as resistant modifiers. Now, onto the problem statement. As stated before, due to the irrational usage of antibiotics, regular antibiotics may be ineffective. Due to a scarce of effective preventative strategies, it is critical to develop a novel therapeutic alternative as well as antimicrobial medications. Now, why is this research significant? First, it will be beneficial to researchers as it may assist to keep antibiotic resistance among bacteria at bay. Not only that, guava leaf have a lot of therapeutic advantages and also is readily available as it can be found abundant all over the world, making this research valuable to society. Carry on. The objective of this review is to discuss the potential antimicrobial property of guava leaf extract as well as to study the effect and properties of the secondary metabolite found in the guava leaf. Let me bring you into the content of the review. Firstly, I want to highlight the overview of Psidium guajava. Guava generally is a tree or a bush which can grow from 1 to 10 meters in height. The fruit could be spherical or pear-shaped and weighs anywhere between 25 to 500 grams. The leaves are broad, bright green, and have prominent veins. Next, onto the use and benefits of guava leaf. Guava leaf have been used throughout history since the Peru civilization cultivated it in the 800 before Common Era. It is commonly infused with water to make tea, but it could also be consumed fresh and also be processed into many different products. Guava leaf have a strong properties in bioremediation. The flavonoid found in the leaf have strong antioxidative impact and also anti-toxicity against aluminum. Not only that, the flavonoid could also be used in the cosmetics and dermatological industry. We can use the flavonoid as formulation. When this antioxidant applied to the skin, it is an essential method to prevent stress oxidative damage. Traditionally, people in India, China, Pakistan, Bangladesh use guava leaf to treat dysentery, flatulence, gastrointestinal problems, diarrhea, and many more. This could trace back to the secondary metabolites found in the guava leaf. These are the examples of the plethora of medicinal application of guava leaf extract, which includes in dentistry, cancer prevention, and etc. Now, onto antimicrobial activity and resistance mechanism. Here are the general mechanism of resistance by microorganisms that explains how they resist medication. In general, for bacterial resistant mechanism, it is divided into 4 different categories. One is drug uptake limitation, second is drug target modification, third is drug inactivation, and fourth is active drug efflux. For viruses are well known to rapidly undergo gene mutation with every successive replication. This increases the chance of resistance to existing antiviral treatment. This is also almost similar to fungi, where the development of resistance is divided into 2 categories, which is the genomic changes and the metabolic changes, which lead to tolerance. To study the antimicrobial properties of Psidium guajava leaf, we must look into its chemical compound and composition. The most prominent and found in copious amount includes saponins, tannins, quercetins, and triterpenoids. Quercetin could increase the solubility and interaction with the bacterial cell membrane, which damages the cell membrane and hindering matrix production. Quercetin could inhibit endocytosis by inhibiting phosphatidylinositol 3 kinase. It can also inhibit the viral genome transcription by inhibiting the RNA polymerase. It could also increase viral clearance by enhancing the mitochondrial antiviral response. Triterpenoid and saponin also have the same properties, but triterpenoid could also inhibit ATP synthesis by inhibiting glycolysis, fatty acid synthesis, and etc. Tannins are plant polyphenolic compounds which binds to the protein, amino acid, alkaloids, and precipitate them, making it an efficient antimicrobial compound. Tannin also have iron chelating properties which inhibit microbial growth by reducing iron absorption by microorganisms. Tannin could also inhibit fungal growth through a variety of mechanisms, which include disruption in micrometabolic activities via inhibition of oxidative phosphorylation, deprivation of essential compounds responsible for microbial growth, and inhibition of enzymes required in the extracellular cytoplasmic membrane. Now, this is the references used in this presentation. In conclusion, Psidium guajava leaf have shown and proven antimicrobial properties, establishing its efficiency in the treatment of the world's most prevalent diseases, making it an imperfect alternative to antibiotics. Guava leaves are highly versatile and highly sustainable with multiple properties. The leaf's application include food, bioremediation, cosmetic, and plethora of other possibilities which owes to the properties of the secondary metabolite found in the leaf. Now for my recommendation of what lies ahead based on the objective. More studies should be conducted as guava leaf are proven to have antimicrobial properties. The higher the quality and quantity of the studies conducted, the more the knowledge that specialists could gain in order to stagnate antibiotic resistance among bacteria. Lastly, more research need to be conducted on the secondary metabolites of the guava leaf. Thousands, hundreds, or maybe thousands of secondary metabolites found have unknown properties which may be beneficial to society. Thank you.